The last hour we'll talk about translation, protein synthesis. And this is a very remarkable process because, I mean, how do you translate from one language to another language? From the language of nucleic acids to the language of proteins. I mean, this is a remarkable process. In each of our cells, we have a translation apparatus that is doing this translation and doing it very accurately. So this is sophisticated and very beautiful. And the overall error rate is maybe 1 error in 10,000 amino acids incorporated. Dictionary, the genetic code, that is the universal translation of our genomes. And of course, this is a very remarkable thing, saying that, I mean, you can summarize to say that all living species on our world are related. I think Darwin was correct when he said we have all evolved from one cell or very few number of cells and getting the same language and also the same machineries, because most of the most complicated machineries, like the translation apparatus, is the same for all species on the world. There are some slight differences, and the RNA polymerase is also very highly conserved between all species. So this, you know, and we don't need to spend any more time than saying that this is a remarkable thing. So how is the translation performed? And I remind you then again of the Crick adapter hypothesis. We have the messenger RNA that needs to be translated, and he was saying that we need an adapter here making the base pairing with the messenger RNA, and we know that the code is a triplet code, so 3 bases in the messenger RNA form the codon, and they are read by the anticodon of the adapter. And in some end of the adapter there is the amino acid. And I showed you yesterday that this is the tRNA, and the amino acid, of course, will be incorporated with the growing polypeptide. And I showed you also yesterday the cloverleaf structure of the tRNA, which is forming in 3 dimensions a beautiful structure in the shape of an L, where the anticodon is up on top here, and the amino acid is attached on the other end. And this is a remarkable shape. All tRNAs look the same, and the 2 business ends of the tRNA are as far apart as 75 angstrom. That says that the ribosome will have to be constructed in a matching way to do the decoding of the message in one end, and then the protein synthesis in the other end. So the ribosome must be built to fit with this. So an important ingredient here are the tRNA synthetases. And Crick suggested that there are at least 20 adapters, 1 for each amino acid, and they would be small RNA molecules, the tRNAs. But how is the amino acid attached correctly? Normally a cell has then 20 charging enzymes, the tRNA synthetases, AARS. And they have a very large variation in size and aggregation. But is there any general theme between them? Because they are dealing with the same type of tRNAs. The tRNAs look the same, all of them. So the synthetases, why are they so different? Is there something common between them? So let's go into the process itself here. The amino acid is then combined with an ATP molecule to this become an activated form of the amino acid. It's becoming attached to an AMP molecule, and the pyrophosphate is dissociating. So this is the activation step performed by the amino acid tRNA synthetases. Then the amino acid AMP is meeting a tRNA bound to the synthetase also, and then the amino acid is transferred to the tRNA, and the AMP molecule will dissociate. So 2 steps of the reaction, and the amino acid has to be correct for this reaction, and the tRNA also has to be correct. And because this is a tricky task, because the amino acids are sometimes very similar in structure, and the tRNAs are also very similar. So you need to identify specifically both in some clearly defined way. Now, the tRNA synthetases, lysine, isoleucine, valine, and so on, they are forming 2 classes. Once we started to get the structures of the tRNA synthetases, then it was very obvious. There are 2 classes, 2 big families. There are 10 amino acids for each of the 2 classes. They have very different structures, but within the class you can see the relationships. And actually the division had been done already before, but people didn't know, people didn't understand that the tRNA synthetases belong to 2 families here. And one family is putting the amino acid on the 2' hydroxyl of the tRNA, and the other family is putting it on the 3' hydroxyl of the tRNA. So there was already the proper division, but one didn't know what the structures were. So in class 1, there are mainly monomeric enzymes, but there are also 3 which are dimeric. So, in class 2, they are usually dimeric, but sometimes they are tetrameric, and there is for glycine there is alpha beta. So it's a heterotetramer, and also for tryptophan there's a heterotetramer, and for for phenylalanine there's a heterotetramer. For alanine there's a homotetramer. So you can divide the amino acids into the non-polar class. They are here, and then over here you have the charged amino acids, and over here you have the aromatic. And somehow there's some relationship between the enzymes. The characteristic differences between the 2 classes of tRNA synthetases is, you can find it in the sequence motifs. The class 1 has 3 different sequence motifs which are normally found. Class 2 has different type of sequence motifs. Then you can divide the classes into subclasses. Subclass 1A or 2A and 1B and 2B and 1C and 2C. So the aromatic, they belong to 2C, and the charged amino acid residues and also the glutamine and asparagine, they belong to the B class, but the hydrophobic generally belong to the A class. And here, as you see, class 1 puts the amino acid group onto the 2' hydroxyl, and class 2 onto the 3' hydroxyl. And what distinguishes the 2 classes more than anything is the fold of the ATP binding domain, because class 1 has the Rossmann fold with a parallel beta structure, but class 2 has an antiparallel beta structure, so they have no relationship between them. But all the enzymes in class 1, they are related, and all the enzymes of class 2 are related. And I may return to that tomorrow. And the amino acid binding here is on the surface, but here it is in a deep pocket. And the acceptor end, the tRNA is here bent, but here it's straight in the complex between synthetase and tRNA. And the tRNA recognition is primarily in the minor groove here of the tRNA, but here it's in the major groove. So there are distinct differences. So this is the shape of some of the tRNA synthetases. Here's one from class 1b. It has 1, 2, 3, 4 domains. So this is the glutamate tRNA synthetase. This is glutamine, which should be fairly similar. And you can see that the blue domains, they have some similarity. The green domains are similar. The purple domains here are not very similar, but maybe partly. But then there's an extra domain here, a pink domain, which has beta structures, and in the same place there is just some helical structure, and the C-terminal domain here is folded by just beta strands, but here is just helices. So they have similarities but differences. So they both 1b, class 1b. But aspartic acid belonging to class 2b is very different. So here you see the antiparallel beta structure in the active site, and big domain, and the other domains have no relationship really to the others. So here we see then the case of aspartate tRNA synthetase. So this is a dimer, and you see here the charging domains in green, and you have the yellow domains here, and there are two tRNAs bound to the enzyme in this beautiful way. And here the anticodon is recognized by the enzyme. So you see here the anticodon G34, U35, and C36, which are hydrogen bonded specifically to parts of the protein. So there is a unique recognition of the anticodon of the aspartate tRNA. And then again, you have to recognize the amino acid very properly, and here is then the amino acid binding site for tyrosine. So this is tyrosine tRNA synthetase. So here is the tyrosine, and if you look into the details, tyrosine is sitting here, and it has an amino acid, hydrogen bond acceptor and hydrogen bond donor to the hydroxyl group. This is exactly what is needed. If you would have two donors or two acceptors, it wouldn't work. But hydroxyl has a free electron pair, and it has a proton, so it will require exactly this. And the proper distance from the alpha carbon and the amino group here, amino group is also recognized, orienting the whole thing in the proper way. So it could only fit for tyrosine. So now, to make sure that you have done the right thing, there is some editing mechanism, and the structure is having an extra domain in some cases, which is hydrolyzing off a wrong amino acid. So the tRNA synthetase needs to identify both the tRNA and the amino acid. In some cases, this is difficult. Leucine, isoleucine, valine, and threonine, you could say, have similar editing domains, and the charging is checked, and the errors are hydrolyzed. So here is then, say, the anticodon recognition domain, this being the tRNA in gray here. Then you have the catalytic domain, which is putting the amino acid onto the tRNA, and then you have the editing domain, which is checking that you really have got the right amino acid bound to this tRNA. And if not, the amino acid will be removed. So here you have a simple illustration of isoleucine, which has the extra methyl group here. It couldn't easily fit into the binding site for a valine, but in case you make mistakes, then you better find out how. And actually, you can say this is a double sieve mechanism. There are two sieves. You're sieving the grains if you say so, by two different sieves. So the first one here is mainly just checking for the size, you can say. And isoleucine here will not fit into the site which is made for valine. So valine is the correct one. It will lead onto the ribosome with the tRNA. But isoleucine is too big, it can't enter. But threonine has very much the same size as valine. So it would come in here, and if you then, by this valine tRNA synthetase, form a threonine AMP, it will be checked and fall off, be hydrolyzed. But in case you add a threonine to the tRNA valine, then that will also be checked and hydrolyzed away. So two sieves, first sieve and second sieve. So here we see the tRNA synthetase for leucine or isoleucine. They're fairly similar, but not identical. You can see this domain is different between the two, and here's the tRNA sitting. So this blue domain is the editing domain. So when the reaction, addition of the amino acid has been done here, it's moved over to the editing domain to check that it is really the correct one. And then if it's the correct one, the tRNA is released and can now engage in protein synthesis. Otherwise, the amino acid will be hydrolyzed off. So the translation now is performed on ribosomes. And in the 70s, when I started to work in this field, the ribosome looked in this way. There is a small subunit and a large subunit. They have some shapes, but basically this is so-called blobology. We're drawing the molecules as blobs, but now, of course, we know that these molecules have lots of details, and the details are important for the function. So the messenger RNA is translated on the ribosome to specific proteins. And the ribosome is a multi-component system. And here I've listed some. So in bacteria, we have the full ribosome is called the 70S particle, and they have small subunits and large subunits. Both subunits have both RNA and proteins. In bacteria, the small subunit has a 16S RNA and 21 different proteins, and the large subunit has both 23S RNA and a 5S RNA and 33 proteins. Chloroplasts are very much the same as bacteria. Archaebacteria are slightly different. Eukaryotes are bigger, have bigger RNA molecules, more RNA molecules, and many more proteins. Mitochondria, the plant mitochondria, they are not really fully investigated yet. Yeast mitochondria, they are lacking the 5S RNA, but they have a little bit smaller RNAs than bacteria, but more proteins. And mammalian mitochondria, they have a lower acidomation constant, but they have much shorter RNA molecules, 12S and 16S, but a very large number of proteins. And trypanosomal ribosomes from mitochondria are the smallest ribosomes. They have the smallest RNAs, but a very large number of proteins. So there is a large variation between various ribosomes, but as I will show you tomorrow, there is also a great deal of conservation. Now we come to the structural work on ribosomes. And Ada Jonat, she was the real pioneer, and she said it must be possible to crystallize also ribosomes, even though they are very diverse in size, and they bind various proteins and drop other proteins off. So these were her first crystals of ribosomes that she could produce, and this was starting already in 1980. So she had a very long road before they got any useful results, but they got there. And this is the other champion, and he is a star crystallographer, you could say, and he was the one to really find the way how to do the complete crystallography on ribosomes, Tom Stites. And I can show you some of the elements of his success. So back in 1999, he was able to produce something, a very low resolution structure from crystallography of the large subunit. And if you compare this to the electron microscopic picture, you can see that it looks the same. This is roughly the same resolution, but there are more details in this picture. But this was 1998, and then the same year he could produce a 9 angstrom resolution structure, and now you can see the details are getting much finer. Then 1999 he could produce a 5 angstrom structure, and here you can see just a part of that structure, how the nice double helical RNA in some part of it here is looking the way it should. And just year 2000, he could arrive at 2.4 angstroms resolution. And now you can see the effect of resolution going from this to that, to, all the way through here. And now you can see the phosphate, the high density of the phosphates all along the chain here. You can see the base pairing along the double helical RNA. So now we know the details, essentially all the details of the ribosome. So here is then a whole ribosome, 30S, the small subunit, and the large subunit. And the spaghetti here, or whatever you would call it, that's the RNA of the small subunit and RNA of the large subunit. And the helices here and the darker blue, these are the proteins, ribosomal proteins. And the purple proteins here, that's the large subunit proteins. Between the two subunits, you see the 3 tRNA molecules, the A site, the P site, and the E site tRNAs. And I'll try to show you how the whole operation takes place. But first, let's have a quick look at the two subunits. The small subunit, the RNA of the small subunit is composed of 4 domains. There's the big body here, and there's the head of the molecule, and there's a shoulder over here. And then the blue stuff is the 5' end of the ribosomal 16S ribosomal RNA, which is just a long helix sitting on the side here. And the active site for decoding is right around here. But these domains say that this small subunit is flexible. So particularly the head can move because there's just one helix connecting the head to the body. So conformational changes are possible between the domains. But if you look at the large subunit, the RNA here is composed of 6 different domains, but they are interwoven with each other in a very complicated way. So this is a stiff domain. You can't do much to it, except that there are proteins on the surface here which are very flexible and have function as flexible proteins. So this again shows the whole thing. I don't need to show that again. So this shows the ribosomal RNA from the small subunit. And you see the 4 domains, the blue, the purple, the red, and here the final, 3' domain with the long helix. But if you go to different species here, from bovine mitochondria, you can see that the red parts, they are now missing because this is a much shorter RNA, but nevertheless, the main features remain the same. So long helix here, and you can make an outline, but just removing certain features. The red features here don't exist in the bovine mitochondria. But in general, you can just say the core is always the same. And this is true for all species. The core of the ribosomal RNA is always the same, but you can add things or you can remove things. Then again here, the ribosome cannot do it by itself. It needs factors, and these are translation factors then. So initiation, some funny thing happened there, has in bacteria 3 factors, but in eukaryotes there are many more factors involved. Elongation, there are also 3 factors, and termination and recycling, there are 4 factors, you can say. So IF1, IF2, IF3, and IFTU, IFTS, IFG during elongation, and release factors 1 and 2 and 3, and then what is called the ribosome recycling factor. Of course, nowadays recycling is a very important phenomenon, but the ribosomes have known about it for billions of years. They have always had a recycling effect because you cannot just waste a ribosome that has been used once. You have to use it over and over again. And as I've shown over on the right-hand side here, there are some of these factors which are GTPases. Previously we have talked about ATPases, which hydrolyze ATP to do the job, but these are GTPases, and there's a slight difference between ATPases and GTPases. ATPases, they are usually motor proteins. They make work. But the GTPases, they are switch proteins. They go from one state to another state. So, and maybe they do some more work also, but generally these are molecular switches, it's called. And the ATPases are motor proteins. So this is the process of translation, and we start over here with the 2 subunits, small and large subunit. And during the initiation here, you first bind a messenger RNA to the small subunit, and this is then able to pick up a formylmethionine tRNA with a formylmethionine bound to it, and then you bind the large subunit to the whole thing. So then you have you come to this state because the formylmethionine tRNA is bound to the P site here, the middle site. But now you're able to start the elongation phase. Actually, the initiation is assisted, of course, by the 3 factors, and we don't have the time to go into all of them. But elongation is maybe the most important. So now in the first step here, there is a protein called elongation factor TU, which actually binds the tRNA and the GTP molecule. So it forms a ternary complex. And actually, as soon as the tRNA is amino-acylated, there is lots of TU molecules in the cell. So all the tRNAs, the charged tRNAs, they are bound to different TU molecules. I think it would be a waste of energy and so on to let the tRNAs, charged tRNAs swim around, so they're bound to the protein to be stabilized and not lose the amino acid. Yeah, in any case. So elongation factor TU with GTP binds amino acid tRNA and brings it into the ribosome here. And when it's on the ribosome here, and I'll show you the details later, the growing peptide is moved from this P site tRNA to the A site tRNA. So it becomes this. So now the A site tRNA has the growing polypeptide on it, and we have an empty tRNA in the P site. So then comes the translocation step, which is catalyzed by elongation factor G. And now that is pushing the tRNA with polypeptide on it from the A site into the P site. So this is done by an interesting mechanism. And then once you're back to this state again, you just go around here for as many cycles as there are codons, proper codons in the gene. But when you hit a stop codon, you know, there are 3 stop codons, then there's no tRNA that would bind to the stop codons. So then you're looking for something else. And actually the release factors here, they recognize the stop codons. They can read the stop codon. And then they hydrolyze the growing polypeptide from the P site tRNA, and release factor 3 with GTP will kick them from the ribosome. You just have a ribosome with a messenger RNA and empty tRNA sitting in the P site and attacked by recycling factor RRF together with elongation factor G, the same factors, to break apart the whole ribosome so you get back the subunits and you can reutilize the whole system again. So that's the complete story, and now I'll show you some of the details. So this is elongation factor TU. Actually, with GDP bound, it has a very open structure, but with GTP, it has a very different structure. So the G domain here gets reoriented with regard to these 2 domains. And it's only in this form with GTP that it can bind a tRNA. So the tRNA is bound here, and there's very little contact between the protein and the RNA, but stable enough, and the amino acid is tucked in here between all 3 domains, actually. So this is a molecular switch, as I've told you already. Now, this binds to the ribosome in a very elegant way. So here is a large subunit again, a small subunit. Here's the P site tRNA, and here's the peptidyl transfer site. So this is the active site on the ribosome, on the large subunit, where the amino acid is getting connected to the growing polypeptide. So the decoding area is down here on the small subunit, but the active site is here. So the 75 angstrom distance between decoding and amino acid is explained by this arrangement here of the ribosome, the large distance between the 2 active sites. Now, when the elongation factor TU comes in with the tRNA, then it has to be tested. And the TU is the part of the system that binds very strongly and happily here. But you can see that this is a wrong conformation. I mean, the tRNA cannot be here if it should be bringing the amino acid to the polypeptide here. So this is a strained and first stage of the system. And actually, the tRNA does not reach the codon, but it needs to make a bend in the the anticodon part needs to make a bend to be tested against the codon. So the tRNA bends and is tested against the codon, and then if it is correct, then it is accepted, and the GTP molecule of FTU will be hydrolyzed, and then, as I say, FTU is a switch protein, molecular switch, so and loses the grip of the tRNA and of the ribosome and falls off. And then releasing the tRNA, this can now stand upright, and place the amino acid right in the active site here and participate in peptide transfer. Here is then the initial binding of TU with the ternary complex. It goes into something called the A/T site. So this is not the proper A site. The other 2 tRNAs are sitting in the P and E site, but this is sitting in the wrong site. Here's the decoding, and up here is the peptide transfer site. So here we can see the difference in conformation between, well, here's the elongation factor TU, and in the ternary complex, aside from the ribosome, or when it comes into the ribosome, it has the bluish conformation, but on the ribosome it makes a bend here. So it reaches the codon. It can read the codon. In this conformation, it cannot read the codon, but here it can read the codon. And this is displaying the dance that the tRNA is going through. In the A/T site, it has this conformation. In the A site, it has this conformation. In the P site, it has the green conformation. And in the E site, it has the yellow conformation. So there's big swings. If you hold on to the codon here, the rest of the tRNA is moving very much. Well, now we come to Venki Ramakrishnan from Cambridge, and his main contribution that was leading to his Nobel Prize 2009 was that he understood how the mechanism is done to identify the correct codon-anticodon interaction. And we can say this was Ramakrishnan's ruler, and I showed you a little bit yesterday about it. So here the UUC is matching the AAG, and there are systems here to measure the distance between the riboses of this A and this U and this A and this U. And I showed you that before. So only if you have the correct Watson-Crick for the first and second base pair here are you accepted. Otherwise, you'll fall off. And some other complex, another tRNA will be tested, because you understand there are so many different tRNAs in solution, and only some of them are correct. So you have to test one after the other, and all the wrong ones will fall off. But as soon as you hit the right one, you better get hold of it and now proceed with the whole process. So this is the system that he made clear. So this is then part of the ribosome. The ribosome is in gray, just a little bit, and the messenger RNA is this purple stuff, and the tRNA is this thing here. So here is then the codon, which is interacting with the anticodon up here, making sometimes the proper Watson-Crick base pairs, sometimes not. But the base is adenine 1492, adenine 1493, and guanine 530. They participate in measuring the distances between Watson-Crick base pair, because if you have a non-Watson-Crick base pair, the distance between the riboses will be different from when you have Watson-Crick. If you go into the details of base pairs, you can see that there's a difference in distance sufficient. So here is the way adenine 1493 checks the first base pair here and measures the distance between the ribose here and the ribose there, making the proper hydrogen bonds. If that's all right, yes, we are on the right way. But if also this second base pair of the codon-anticodon is correct, checked by these parts of the ribosome, then you're okay. The third base pair is not so important, but of course it is also checked to some slight extent. And that's called the wobble base pair, which is. Now, we come to the stage where the molecular switch is being switched, and that is the GTP hydrolysis. And this has been a problem for a long, long time. How is this done? And the elongation factor TU that binds the GTP here has a residue, a histidine residue, but it seemed to be too far away to do any proper job at all. But now a group made the experiment where this histidine was in an activated state, and now it was moved in close proximity of the GTP. Here you see the alpha, beta, and gamma phosphates of the GTP, and here's a water molecule sitting. So the histidine is pushing the water up against the gamma phosphate, and now through elegant reactions which have been very thoroughly investigated by say, computational models, this water becomes an hydroxyl and attacks the phosphate here, and the proton that is lost is moving over to this phosphate, and then the bond is broken here. So you just have a GDP, and with GDP, as I told you, elongation factor TU flies. This is the touch with the ribosome and the tRNA. So let's proceed. Now elongation factor TU with GDP falls off, and now the amino acid stands upright here, close to the brings its amino acid into the peptide transfer center. And this shows another feature of the ribosome. So here's the peptide transfer center, but through the whole large subunit is a channel through which the protein is coming out, and it's about 100 angstrom in length. And the protein is protected from aggressive proteases out in the solution for the whole length. So it'll be something like 30, 40 amino acids which are shielded by the ribosome before they come into the outer world. So the peptide transfer center is right here. Now, so this is the A site tRNA, which has now been allowed to stand upright here and brings its amino acid next to the peptide. So this is the B site tRNA with the polypeptide, and here's the exit channel for the growing polypeptide. And I haven't mentioned this before, the ends of the tRNAs here, the acceptor ends, they have the sequence CCA, and all the tRNAs have this CCA, and that is matching very perfectly with two guanines of the ribosome, which makes base pairs and holds it right into the proper position, because otherwise catalysis wouldn't be able to happen. And also the A site tRNA has a guanine here making proper Watson-Crick base pair, holding this C in the proper place. And there are additional interactions with the adenines here also, which makes the whole thing stable into the active site. And then all of a sudden, the whole peptide is moved over to the A site tRNA. So now you have a peptidal tRNA sitting in the A site and an empty tRNA in the P site. And it's quite interesting to notice which groups are participating here. There's a nitrogen of adenine 2451 in the large subunit, which is important in the hydrogen bonding of the amino group that is attacking here, the carbonyl carbon. But another group that is important is the 2' hydroxyl of the P site tRNA. So the 2' hydroxyl is participating in the catalysis here. So this is something which is called substrate-assisted catalysis. The substrate, the tRNA in the P site, is assisting the catalysis here. And one is saying that reactions which are substrate-assisted catalysis, they are very ancient reactions. And of course, this is a very ancient reaction. As long as we have had proteins, this mechanism must have been in operation, I think. So here you see the base pairing of the C residues of the tRNAs. Okay, so now we're coming to the translocation step. Now we have made the peptide, so we need to push the tRNA over from A site to the P site, and this is done by elongation factor G, and this is the way it looks. It has 5 or even 6 domains. It's a large and complicated protein, and our group determined the structure 20 years ago, or 21 years ago, because that was a happy moment. So here's the GTP molecule sitting. Now, I think you've seen this structure before, because elongation factor TU with tRNA is very much looking alike. So this part of elongation factor G is a tRNA imitation made in protein. And this was the first time that anybody had seen such an imitation done. And if you look at the next picture, we can see how similar they behave. Oops. So here is what you seen already, elongation factor TU coming in, but now elongation factor G comes into the same site and brings its tRNA imitation right into the tRNA binding site. So, of course, the tRNA cannot sit here when this part of the protein comes in, so it's being pushed over. So this is the way it happens. Oh, yes. Here we go. So just a simple, stupid representation of the whole thing. So this illustration shows here the P site tRNA, and it shows the A site tRNA in just faint gray, and this the red stuff here is elongation factor G, domain 4, which is the part that is coming closest to the tRNA binding site. So here's the messenger, and so to say, the A site tRNA cannot remain here if elongation factor G comes in. And then we come to the final bit, which is the release, and let's do it quite rapidly. So this is the release factor 2, and the crystal structure of this was looking beautiful and interesting, but there is a problem, because one has identified here that there's a sequence here called serine, proline, phenylalanine, which is the part that goes closest to the stop codon, and the GGQ sequence, which is the one participating in hydrolysis of the amino acid, is sitting only 23 angstrom away. So this cannot be, because they should be 75 angstrom apart. But when this was the crystal structure of the factor alone, but when you look at it sitting on the ribosome, there's a large conformational change of the protein that now brings the distance between these two sites to 73 angstrom. So that would be proper and working. So here's the release factor 2 sitting on the ribosome next to the P site tRNA, doing the hydrolysis over here, reading the stop codon over there. And then in the next step, release factor number 3 binds to the GTPase binding factor site and kicks the release factor 2 off. And then we come to the very last bit here, which is the ribosome recycling factor. And this was also done by my group, Maria Selmer in my group. And the blue structure is the ribosome recycling factor. But she observed that this looks very much like a tRNA also. So we fell into this observation several times that a protein looks like a tRNA. And we thought maybe it binds also to a tRNA site. But this turned out to be wrong. Because this is the way the factor binds. So here are the 3 tRNA binding sites against the small subunit A site, P site, and E site. But ribosome recycling factor binds in quite a different way. But certainly it can perform its job also in this conformation. So the ribosome recycling factor sits in this way on the ribosome, and elongation factor G comes in and then splits apart the 2 subunits from each other, and has then produced the recycling. I think I'll finish by trying to show you this movie, which is from Venki Ramakrishna's group, and which summarizes some of the stuff. So here we have the messenger RNA, and we have the different factors and the small subunit. So now the F-met tRNA comes into the binding site in the P site here, and now the large subunit is docking onto the small subunit, and the initiation factors disappear. This is the initiation factor 2. So now we're ready for elongation. And here comes elongation factor TU with the tRNA bound, and it finds its proper site, it making the bend to identify, and here GTP is hydrolyzed and elongation factor TU falls off. Now the peptide is possible to be made here, and here comes elongation factor G, ready to push away the A site tRNA from the A site into the P site, and then this undergoes GTP hydrolysis and falls off. Here comes the next elongation factor T with its tRNA, and now we get into more realistic rate, because there are 20 amino acids incorporated per second. So even this is a slow version of the whole thing. And here you see the growing polypeptide coming out through the exit channel on the backside and trying to fold in the best way it can. And now we come to termination. So now release factors 1 and 2 recognize the stop codon on the small subunit and hydrolyze off the polypeptide that disappears. And here comes release factor, recycling factor and elongation factor G, and splitting apart the ribosome. Okay, so that's it. I'm sorry for extending the session too much.